안녕하세요. 네. 저 뇌영상센터 이상훈이라고 합니다. 반갑습니다. 네. From now on, we'll proceed in English. Please understand. So, good afternoon, and I thank you all for coming to the first talk of this year's colloquium at SNU Brain Imaging Center. I'm Saman Lee, from the Department of Brain and Cognitive Sciences. I also serve as director of the center. It is my great pleasure to welcoming Professor Michael Silver, today's speaker, of University of California, Berkeley. Professor Silver is a leading figure in vision and cognitive sciences. He's currently a professor of UC Berkeley, at optometry and vision science, right? And also professor of neuroscience. And he also serves as director of the interesting name center, UC Berkeley Center for the Science of Psychedelics. And he completed, briefly, I talk about his education background. He completed his undergraduate studies at Carnegie Mellon University, earning a dual bachelor's degrees in chemistry and biological sciences. And then he received his PhD in neuroscience from UC San Francisco, where he trained with Michael Stracker. Is that correct? That Michael Stracker. He then pursued the postdoctoral training at the Riken Institute in Japan, studying the effects of nerve growth factor on cortical EEG activity, and later worked at the Max Planck Institute with Nikos Logophilis on psychophysical and electrophysiological studies of the neural correlates of visual perception. This strong interdisciplinary foundation is reflected throughout his career, which embraces physiology, chemistry, psychophysics, computation, modeling in systems, neuroscience. His research spans an impressive range of approaches from psychophysics, neuroimaging to pharmacological manipulations, all aimed at the understanding the cognitive neural mechanisms, how it gives rise to perception, attention, and learning. And throughout the years, his work has made fundamental contributions to our understanding attention, perception, learning. But also he did it through the elegant combination of fMRI and computational approaches in pharmacology. And so today he will speak about the role of acetylcholine in the human visual cortex. And I believe in today's talk, he will describe how pharmacological enhancement of cholinergic function using the Donepezil, is that also correct pronunciation? Perfect. The Donepezil. The effects of perceptual learning and visual attention and encoding of visual information in the cortex. His findings provide evidence that increasing cholinergic transmission can mimic key effects of attention, such as sharpening neural cell activity and facilitating learning. And from now on, this is personal notes, and so he and I were postdocs together, and David Hilger's lab at Stanford and MIT long years ago. And I believe those years are kind of formative years for both of us. And so highly postdoc-centered lab. So one fun time was always afternoon. We postdocs got together and sitting at the coffee table and had a lot of discussions. And I remember that Michael always brought to our discussion the scientific curiosity and energy. I still remember those years, and then it's very meaningful for me to have you here at SNL. So please join me in welcoming Professor Silver.
Thank you very much, Sigurd, for that lovely introduction. And it's such an honor to be invited to give a colloquium here at one of the world's great universities. And I'm really excited to learn more about the work that's happening here. and grateful for you being here today and for the invitation to be here. And also to Young, who was very helpful in arranging my trip and organizing and giving me advice. And I feel very welcome here. So thank you for that. The star of the colloquium today is the neurotransmitter acetylcholine. This is the first neurotransmitter ever to be discovered, and surprisingly recently. So about 100 years ago was the discovery of acetylcholine, and all neurotransmitters that we know of have been discovered since then. So everything we know about the action of neurotransmitters, receptor types, the development of pharmaceutical drugs based on that knowledge, it's all in the last century. So an incredible amount of knowledge in a very short amount of time. And acetylcholine is a neurotransmitter that has many different kinds of effects. So it is the single transmitter at the neuromuscular junction, the synapse between motor neurons and skeletal muscle. It's also a transmitter in sympathetic and parasympathetic neurons within the autonomic nervous system. And of course, it is a major neurotransmitter in the central nervous system. So this shows the major projections of the cholinergic or acetylcholine neurons in the brain. And so there are multiple nuclei in the brainstem that are very involved in the regulation of sleep and wakefulness and are very active during REM sleep in particular. And there are also a cluster of neurons in the basal forebrain that project widely over the cerebral cortex. And these projections are extremely diffuse. So a single cell can project to multiple brain areas, even in multiple lobes of the brain in the cerebral cortex. So very broad, diffuse anatomical projections from the basal forebrain cholinergic nuclei to the cerebral cortex. There are many drugs that interact with the cholinergic system. Maybe most famous is nicotine, which is the active ingredient in tobacco, cigarettes. It acts at one class of acetylcholine receptors called the nicotinic receptors, named after nicotine. The other class is muscarinic receptors, and there are multiple drugs that interact there, including atropine and scopolamine. And then there are many drugs that have potentially life-threatening effects on the cholinergic system. So forms of nerve gas or the venom in black widow spiders, the toxin that botulinum bacteria make, these are all acting on the cholinergic system and can be lethal because of the cholinergic system's important roles in regulating heart rate and enabling skeletal muscle to contract. And then there are other medications that are beneficial for human health. And these are typically used to treat Alzheimer's disease and related dementias. So these are cholinesterase inhibitors. They block the enzyme that normally breaks down or inactivates acetylcholine in the synapse. And some of these drugs are physostigmine, rivastigmine, and one that we'll talk about a lot today, donepezil, also known as trade name Aricept. And so these are very useful scientifically because these drugs, because they prolong the lifetime of acetylcholine in the synapse, in a sense, they are mimicking the normal physiological functions of acetylcholine, but they're enhancing those. And so it's a way of understanding the basic biology of acetylcholine. And so their function, because they block the enzyme that breaks down acetylcholine, when it is released into the synapse, stays in the synapse for a longer period of time. It's also really helpful for research purposes that this is the most common treatment for Alzheimer's disease. And so pharmaceutical companies have invested a huge amount of research and money into developing better and better cholinesterase inhibitors that are more specific to the brain, that have fewer peripheral side effects, that can be well tolerated, that are orally active. And so all of these are very positive aspects of the drugs for use in research purposes, not with patients necessarily, but also with neurotypical or so-called healthy normal people. And part of what's interesting about this drug in terms of cognitive neuroscience is that donepezil increases learning and memory and attention in patients with Alzheimer's disease. And so this is just a movie to show the molecular action of the drug. So here we have a presynaptic terminal of an axon with synaptic vesicles containing acetylcholine in blue. Of course, when an action potential arrives, these vesicles fuse with the plasma membrane and release acetylcholine into the synapse. And there, acetylcholine interacts with cholinergic receptors on the postsynaptic side of the dendrite. And there is a naturally occurring enzyme called cholinesterase. It's one of the fastest enzymes, actually, of all enzymes. And it very rapidly converts acetylcholine to an inactive form. And this is very helpful because now when acetylcholine is released, it signals to the receptors and then is immediately inactivated. So subsequent action potentials are distinct and separated from the previous release of acetylcholine. And donepezil and drugs of that class bind to this enzyme and inhibit its activity. So it cannot function as well anymore. And so now when acetylcholine is released, it stays in the synapse longer. There's more time before it's inactivated. And so really think of it as just increasing the gain of the cholinergic system, because any synapse that releases acetylcholine will have a potentiation of the cholinergic effects. It's not, unlike drugs like nicotine or scopolamine, it's not specific to a receptor subtype. Whatever receptors are naturally occurring at the synapse will be activated in the same way, but just for longer or more sustained period of time. And we're, in our lab, we're really interested in understanding acetylcholine in the context of attention and learning and the interrelationships between acetylcholine as a signaling molecule and then the cognitive and neurobiological phenomena of attention and learning. And the very first experiments that we did were simply to give donepezil to healthy human subjects and examine the responses to visual stimulation in visual cortex. So I'm sure, as many of you know, visual cortex is organized into retinotopic cortical areas. That means areas that have a full continuous map of the visual field. And it's a contralateral map so that the right side of the visual field is represented in visual cortical areas on the left side of the brain. The left side of the visual field is represented in the right side of the brain. And adjacent locations in the world will illuminate adjacent locations in the retina. And that two-dimensional map of the retina is repeated in the lateral nucleus in the thalamus, in primary visual cortex, area V1, and in many other cortical areas. And so that's why it's called retinotopic, because it is a copy, in a way, of the map of visual space that's first established in the retina. And the kinds of techniques that Sang-Hun and I learned more than 25 years ago at Stanford enables us to you can identify these areas in individual subjects, and not only can you identify the boundaries of the areas, you can identify the map of visual locations. So here we have a flattened representation of the right hemisphere, and so you can see here is the map of visual space. So stimuli of different polar angle are represented with different colors. So the upper vertical meridian, so above the point of fixation, is shown in red, and the lower vertical meridian below the location of gaze is shown here in blue. And you can see moving across, for example, primary visual cortex, it goes from red to yellow to green to cyan. So basically from this upper vertical meridian all the way down to the lower vertical meridian. And these colors here are not represented in this hemisphere. They're the right side of visual space. They'd be represented in the left hemisphere, which is not shown here. And so you can see the continuous map of visual space and also the reversals of these maps at the boundaries between adjacent areas in V2 and V3. This shows the map of eccentricity, so how central versus peripheral a location is. And here you see bands of eccentricity that span across these early visual areas. So these regions here are representing areas close to the central visual field, and then middle eccentricities and out into the periphery. So this is a huge advance in visual neuroscience, being able to use non-invasive techniques like fMRI to be able to map the locations and the visual field layout in all of these different areas, because it enables you to then has very precise hypotheses about the function of each of these areas. And so what do we know about acetylcholine? One advantage of doing pharmacology is that the very same drugs are often used in humans and also in animal models and slices in culture. And so that provides really strong links between different levels of analysis in neuroscience. And so we know from slices of rat visual cortex that putting acetylcholine in the bath, so covering the slice with acetylcholine, reduces the spread of excitatory activity in this slice. So here there's electrical stimulation being delivered, and it produces a certain area of activation. That area is reduced when acetylcholine levels are higher. It's also known in intact animals that locally administering acetylcholine to primary visual cortex reduces the receptive field size. So if you present an optimal bar of light to activate the neuron that's being recorded, and then you vary the length of that bar, there's a best optimal length for that neuron. That optimal length reduces when acetylcholine is present. And so putting these together, the idea is that acetylcholine, based on these circuit level measurements, is limiting the amount of excitatory spatial spread of activation in visual cortex. And so we had that knowledge from the animal literature, and then we looked to see what acetylcholine or increases in acetylcholine with donepezil would do to fMRI responses to visual cortical stimulation. And so we had a very simple stimulus, which is just a checkerboard that is an annulus shape around the fixation point. And this checkerboard was contrast reversing to be able to drive visual cortical responses very well. And because early visual cortical areas have a map of the visual field, some parts of that map will be within the stimulus representation, and some other parts of the map will be either more central or more peripheral. And so we expect that those parts of the map that are excited by the stimulus will show this pattern. If you could record from the neurons would have more activity when the stimulus was on, and then less activity when the stimulus is off. So the stimulus is on for about 9 or 10 seconds, and then off, and then on, and then off. Just a simple block design. And then there might be other neurons that, for example, are representing locations out here that are suppressed by the stimulus coming on. And so those would show the same fluctuation in activity, but it would be out of phase, 180 degrees out of phase. So it would be suppressed when the stimulus is on, and actually higher activity when the stimulus is off. So, of course, we don't measure neurons directly with fMRI. We measure the bold signal. And so convolving this model of the neurons with the hemodynamic response function, we should get a roughly sinusoidal modulation of activity at the period of the stimulus cycle, so 10 seconds on and 10 seconds off. And we should also see maybe a negative bold response, a suppressive response in other parts of the visual map. So the same period, same frequency, but out of phase. And so for every single voxel in these identified visual cortical areas, we determined how much we would need to advance or delay the phase of this sinusoidal model to best account for the pattern of activity in that single voxel. And we expected to find two populations, one showing a positive bold response to the stimulus and one showing a negative bold response. And this shows on a flattened representation of cortex the actual phases. And so you can actually see the shape of the stimulus. Remember, the contralateral organization, of course, means that only half of the stimulus is going to be represented in this right hemisphere. And you can actually see a hemicircle and hemihelix shape on the cortical representation. And so here is the left horizontal meridian in the middle of V1. Here's the lower vertical meridian at the boundary between V1 and dorsal V2. And the upper vertical meridian is at the boundary between V1 and ventral V2. And so I haven't yet told you what the color map is here, but here's, you can see this is now a histogram of the phases. And so you see that there's really a very strong bimodal distribution. In this part of the map that represents the stimulus, you see phases that are consistent with an excitatory response, a positive bold response. But both centrally and peripherally, you see phases 180 degrees away that are consistent with a negative response to the stimulus, even parts of V1 that are very far away from the stimulus representation. So this is many degrees of visual space away. And essentially, all parts of V1 that we mapped out have either an excitatory or an inhibitory response. Almost all of the voxels fall within these two modes. And so we can simply find the boundaries that best separate these and classify each voxel in the brain as either a positive response or a negative response. And so when we do that, we can look at the effects of acetylcholine in a way that is independent of possible drug effects on amplitude or on the signal-to-noise ratio of the response, because every single voxel in V1 is being classified either as positive or negative. And so here's an example from just one person's brain. All the red points are those that show a positive response, and blue are those that show a negative response. And this yellow is simply the boundary between the two for this participant. And when the same subject views stimuli in the scanner after taking donepezil, which is enhancing acetylcholine signaling, you see that the boundary between red and blue shifts inward. And so basically this is very consistent with the slice physiology, showing that the extent of excitatory responses is less when acetylcholine levels are higher. So literally a smaller proportion of early visual cortical areas are excited by the stimulus when acetylcholine levels are increased, and this is separate from any possible effects on amplitude or reliability of the response. So if you were to record with a microelectrode right here, you would find under placebo, these neurons would be excited, but under donepezil, those same neurons would presumably be suppressed. So there's less visual cortical area that is being excited by the stimulus. And we looked at this across multiple subjects. I think these days to do a study like this, we would want more subjects than 5, but we had many sessions per subject. And overall, there's a clear decrease in the excitatory spatial extent of the response in visual cortex. And so given that finding, we can try to relate that to what's known about cholinergic actions at the circuit level. And a classic model of cholinergic function from Michael Hasselmel and colleagues is more in the context of learning and memory. When acetylcholine levels are higher, that is emphasizing the feed-forward or bottom-up responses in visual cortex or other cortical areas, and is suppressing the intracortical responses. And when acetylcholine levels are low, there's more emphasis on these lateral connections within the cortex and less emphasis on the feed-forward connections. And the idea here is that when animals or people are in a learning state and they're really emphasizing collecting information from the environment, the entire brain is going to more of a feed-forward mode of processing. And then when acetylcholine levels are lower, that's more of a consolidation state where there's a lot of intracortical connections and not so much driving by sensory input. And so in our case, the donepezil is like the high acetylcholine, and if it is increasing the feed-forward input from the thalamus, then what predictions would we make about the receptive fields in primary visual cortex? So in visual cortex, of course, there's bottom-up inputs from the LGN, the lateral genicular nucleus, shown in green. The red are the lateral connections within primary visual cortex, and then broader feedback projections from higher level cortical areas. And so based on the Hasselmel model, we predict that the feed-forward input from the LGN is going to be enhanced and that the lateral connections will be suppressed. And the receptive field size in the LGN is much smaller than that in the cortex. So if the entire cortex is shifting towards more of a bottom-up regime, then the receptive fields in the cortex maybe would come to resemble the receptive fields of the LGN more, and they would shrink. And if you think about shrinking receptive field size everywhere in the cortex, you maybe would get a effect like this, where all the receptive fields are smaller, and so at the population level, there's a reduced excitatory spatial spread. So we had this interesting situation. It's so common for, especially in vision science, for there to be discovery of illusions or perceptual phenomena or just careful introspection that then inspires neuroscientists to try to understand how those perceptual phenomena are happening in the brain. And it's very rare that some pattern of brain activity is discovered with unknown perceptual consequences. But that's kind of the situation we have here. We have this reduced excitatory spatial spread, which seems like it's probably smaller receptive fields. And so what would that mean for perception? And at first glance, it seems like it would be good, right? We all want higher resolution cameras. If you have a higher resolution, smaller receptive field representation of the world, you should have more precise vision, maybe better visual acuity. But that's only one part of what vision does. Vision also needs to integrate. So for texture perception or material perception or summary statistics in the environment, so getting the gist of a scene, contour integration, these all require communication across parts of the visual field, and smaller receptive fields might actually be an impediment. So if an epicell is shifting V1 away from the lateral connections and towards the thalamocortical connections, it should help for tasks that require segmentation or fine visual acuity, but it may be detrimental for other kinds of tasks that require more integration. So we started with visual crowding, which is a phenomenon of peripheral vision. So if you all fixate in the center here and don't move your eyes and try to read the letter to the right, it's extremely easy, hopefully. And the same letter on the left is much more difficult because it's flanked by other letters. And so this is a limitation on peripheral vision. It's probably reflecting the fact that peripheral vision is not so much for identifying features with detail. It's more about getting a summary statistical representation of the environment. And it's not unique to letters. So if your task is to determine whether the smile is right side up or upside down, that's easy to do when the smile is in isolation. But if it's embedded in a face in the periphery, then it's difficult. And so we tested the effects of donepezil on crowding, on visual crowding, and we measured the critical spacing, which is what's the distance between a central letter in the periphery and flanking letters that can enable a certain level of performance in identifying the letter. So it's a letter identification task. So these are presented in the periphery, and then there were only 10 possible letter responses, and so participants, in this case, the correct response is C, of course, and we measured using psychophysical staircases what is the target flanker distance associated with threshold level performance. So a smaller critical spacing means better performance. And so, of course, going out into the periphery, critical spacing increases. There's more and more crowding. But we didn't find any effects of cholinergic enhancement at all. And if you think crowding is fundamentally due to limitations on receptive field size, then reducing receptive field size should be helpful. And so this provides evidence that crowding is not so much about receptive field size, it's more about integrating features and combining features in the periphery. And so from that perspective, acetylcholine maybe wouldn't affect the performance of a crowding task. What about another task that involves spatial interactions in vision? This is called orientation selective surround suppression. So here, if you were performing this task, you would need to determine which of the 4 quadrants has a higher contrast than the other 3. And we now changed the difference between the target quadrant and the other quadrants to measure a threshold level of performance. Performance is significantly worse when there is a surround presented, but it depends very much on the orientation of the surround. So a surround that has an orthogonal perpendicular orientation to the central annulus doesn't cause much surround suppression. But here on the right, you see really strong surround suppression from a parallel surround. And so this is really helpful because it really ties the surround suppression computation to early visual cortical areas because it's orientation selective, and those are the cells that most prominently have orientation selective responses. So we measured this surround suppression in the parallel surround, the orthogonal surround, the no surround. Again, this is a threshold measure, so higher levels mean worse performance. And so you see this really strong suppression effect when the parallel surround is there, much higher levels of threshold contrast compared to the orthogonal surround or the no surround. And here we see an enhancement of performance with donepezil. So thresholds are lower, meaning that the performance of the surround suppression task is better when acetylcholine levels are enhanced. And it's kind of amazing that any systemic drug could improve performance, right? This drug's taken orally, it's increasing acetylcholine levels all over the brain indiscriminately. But even so, that is of benefit for performing this task. And here you might think that could be related to receptive field size changes. If receptive fields are smaller, then maybe you have a receptive field under normal conditions that is combining the center and the surround. But under cases of donepezil, the receptive fields shrink, and so you have a better separation in the visual cortex between the center annulus and the surround stimulus. And so this fits very well with the Hesselmo model, and in general, the idea that acetylcholine is making the cortex more feed forward, and so it's inheriting that small receptive field structure from the LGN, and then that facilitates performance. And we did one final task to try to understand this a little more systematically. And so, sorry, this might be quite difficult to see. Basically, there's a target here, which is made of over superimposed faces, and that part's not relevant for the talk today. What's relevant is that there's also distractors, and we varied the distance between the target and the distractors. So that you can see better down here. At a given eccentricity, there's some distance between the target and the distractor. And so these are the different stimulus conditions. So sometimes the distractors were very close to the target, and sometimes they were much further away. And now we can really systematically look at target-distractor interactions over space. And what you find is that the x-axis here is the distance between targets and distractors. Again, a threshold detection task. In this case, a contrast decrement is being detected. And you see that the thresholds are lowest for this sort of intermediate target distractor distance. So there's this kind of dipper function shape. And what we found is that when people perform this task under a placebo or under donepezil conditions, that donepezil again enhances performance, and it does so selectively at these intermediate target distances. And so the right graph here plots just the benefits of acetylcholine. So positive values means that donepezil is enhancing performance, and the greatest enhancement is at this intermediate target distractor distance. And the value of doing it this way is now we can separately model facilitatory effects, so beneficial effects of distractors compared to suppressive effects of distractors. They each have their own spatial scale. And so we can then fit the data using these difference of Gaussian functions. And so we can model the psychophysical data looking at the amplitude of suppression, the spatial extent of suppression, the width, and then also the amplitude and width of facilitation. And we see that the selective effect of the drug is to reduce the spatial selectivity of facilitation. Again, very consistent with the animal literature and the slice physiology that shows that there's a reduced excitatory spread of activity. So starting with animal literature that inspired the fMRI experiments, documenting that there's a reduced excitatory spatial spread of responses with fMRI, and then finding the perceptual consequences of that in these tasks that involve spatial interactions in vision. So these are the three behavioral tasks that we looked at. Cholinergic enhancement had no effects on visual crowding for a letter identification task. It did enhance performance for orientation selective surround suppression, and it improved performance on this contrast decrement detection task in a way that seems to selectively involve excitatory facilitatory interactions. So that's a just basic characterization of the physiology of acetylcholine effects in the cortex and visual perception. And now I'm going to shift to perceptual learning. So this is Ariel Rookham, a former student in the lab. Ariel, when he moved to California, got really interested in local mushroom collecting And here he is undergoing perceptual learning. So he's using his guidebook to identify this mushroom and to determine what visual features it has that are different from other mushrooms. So these are pictures from mushrooms in California. One of these is the chanterelle, which is a delicious gourmet mushroom. The other is the false chanterelle, which looks very similar, but will give you a lot of intestinal distress if you eat it. And so they differ in ways that can be learned. And that's the phenomenon of perceptual learning, is being able to do visual discriminations that you at the beginning cannot do, but with practice and feedback, you're able to retune your brain to be able to reliably make those visual discriminations. And Ariel was really interested in acetylcholine in the context of perceptual learning. So these are just the basic definitions of perceptual learning. It's a perceptual discrimination that's being learned. Interestingly, it's persistent. And so if you learn a task and get better at it, even if you don't practice it, the benefits last for weeks or months or even years. And it's, in some cases, at least specific, meaning that if you learn a task, for example, a color discrimination task in certain colors, that doesn't necessarily transfer to other colors. Or if you learn a particular orientation discrimination task, that doesn't transfer to other orientations. And so some people have argued that that implies an early visual cortical locus for this, that it's the selectivity of the learning is reflecting the selectivity of the neurons that are showing the plasticity underlying the learning. And so those are happening in these early visual areas. Incredibly, you can practice very, very basic tasks like orientation discrimination or motion discrimination. You would think over our lifetime of using our visual systems that we would be very good at these fundamental visual tasks. But the literature is very clear that there's lots of room for improvement. And if you practice again and again over a period of days, you can really show dramatic improvements, even in very basic visual tasks. And so now we're going to look at the links between acetylcholine and learning. And again, we can really draw upon the animal literature of other studies of acetylcholine. So it's known, for example, that tasks that require sustained attention in animals result in increased release of acetylcholine in the cortex. If you lesion the basal forebrain, which is the source of acetylcholine, that impairs performance on attention tasks. It's also known that attention is, at least for some situations, required for perceptual learning to occur. So if you passively show animals or people the stimuli that are used in a perceptual learning task, but don't require them to do any discrimination or attend to the stimuli, you don't see any plasticity or learning. The learning is dependent on them attending to the stimulus. And finally, infusing acetylcholine into the cortex or electrically stimulating the basal forebrain can actually act like attention for inducing plasticity. So if you have a situation where animals are ignoring or passively viewing stimuli, normally that wouldn't produce the plasticity associated with perceptual learning. But if you increase acetylcholine seems to kind of act in the way that attention would. And so now you get these learning effects even in the absence of explicit attention. So Ariel took a classic task from the visual perceptual learning literature. This is a motion direction discrimination task. So the dots are moving within this annulus in a particular direction, and then there are two intervals. And the second interval either has the exact same direction of motion or a slightly different direction of motion within the annulus. And that's the threshold measure for this task, is how different do these two directions of motion have to be for people to be able to reliably distinguish them. And that is the response categories, either were the directions exactly the same or were they different. And this alpha parameterizes the minimum amount of difference in directions that can be detected. So the task was being done on just parts of the annulus. And so this shows the basic direction of dots up and to the right and these two quadrants. Ariel did this because perceptual learning is selective to stimulus parameters. And so you can actually train people on this direction of motion and these locations, and it won't transfer to these directions of motion and these locations. And so now each person can actually do perceptual learning twice in our study, once under a placebo and once under conditions of cholinergic enhancement. And we can then have a very sensitive within subject measure of the effects of acetylcholine on learning, because each person is their own control, basically. And so we tested the learning by looking at all combinations of direction of motion and location. It was a placebo-controlled double-blind design. And in this case, perceptual learning occurs over days. So people are taking Aricept every single day for a period of, let's say, 7 to 10 days while they're training on one of these conditions. And then we wait 2 weeks for the drug to be removed from the body, or in those cases where people are taking the placebo first, we have the same waiting period, and then whatever condition they were in for the first course of training, placebo or donepezil, they're in the other one for the second. So here's the basic design. Each person got either donepezil or placebo, and the opposite in the second course. We then measured the baseline performance of the task. We trained for 5 days, and then we looked at the effects of treating. There are a lot of measurements of the selectivity of learning and so forth that I won't get into today, but basically under placebo, people showed perceptual learning. So even this very basic motion direction discrimination, people can improve with practice. But the amount of improvement is much greater under donepezil. So having, again, just systemic increases in acetylcholine continuously, chronically for multiple days facilitated learning of this task. And we brought these participants back several months later after the task was finished. They hadn't been practicing motion direction discrimination. They hadn't been taking the drug. And you can see the benefit. First of all, perceptual learning of this task is long-lasting, as was already known. But the benefits of the drug on perceptual learning are also long-lasting. And so we're excited about this as a possible treatment for amblyopia or other conditions of vision where there's some problem in development with the visual system that, for example, with the eyes, there's a misalignment of the eyes or cataracts. Even if that's taken care of surgically, there are long-lasting consequences due to cortical miswiring. And so a drug that enhances plasticity in the adult brain could be combined with these perceptual learning techniques to maybe restore vision in the amblyopic eye. We have so far not been successful in doing that, but this is one kind of feature of the drug effect on learning that is really important, that it seems to persist, because you really don't want a therapy for amblyopia that requires people to train every day the rest of their lives or to be on a drug every day. And so having a targeted intervention that enables plasticity, enables rewiring to take place, and then is persistent after that is the kind of therapeutic that we're looking for. All right, now closing the triangle here, we're going to study the effects of acetylcholine on attention. And so I'm sure many of you are familiar with these two fundamental types of attention, voluntary and involuntary attention. Voluntary attention is goal-directed, sometimes called endogenous attention. It's the kind of attention that we use when we're focused on a task and we're trying to resist distractors in the environment. It's generally slow, so a shift in voluntary attention takes a few hundred milliseconds. And it's the kind that we can sustain for long periods of time. Involuntary attention, sometimes called exogenous attention, is the kind that is captured by events in the environment. So even though you're really maybe focused on the slides or my voice, when the microphone made that sound, your attention was grabbed by that. Even if you were trying to resist that, that's an example of a shift in exogenous attention. And it is fast and transient, so it is engaged very quickly, but it also dissipates very quickly. And this is the evolutionarily ancient type of attention that we use to not get eaten by predators and to be able to react almost reflexively to events in our environment. The voluntary attention is a later evolutionary development that's associated with cognitive control and executive function and being able to maintain our attention on a task in the presence of distractors. So in some ways, these two are opposed to each other, and it's really helpful that we have both. We need involuntary attention to be able to react to unexpected events that maybe are dangerous for us. And it's also, of course, beneficial to have cognitive control and be able to employ voluntary attention to do more complex kinds of tasks. And so Michael Posner and colleagues many decades ago came up with a way of separating the effects of voluntary and involuntary attention on perception. And so we adopted this task for looking at the effects of cholinergic enhancement. And so so you probably are familiar with the cueing task of Michael Posner, where there's something that indicates a location is likely to be where a stimulus will appear. And so you can either direct your attention over there voluntarily, or maybe there's a cue that captures your attention involuntarily. Here, everything is is kind of flipped. And so it's called anti-cueing because the cue in one location indicates that the target is likely to appear in the opposite location. So if there's a cue here, for example, the cue is a darkening of this frame and a stimulus onset asynchrony, so an interval of time after the cue. The target, in this case, is a tilted grating in the middle of these three gratings. And so here the target is on the right side, and that's the most common condition. And the participants are aware of this, that if the cue appears on the left side, the target is probably going to appear on the right side. If the cue appears on the right side, the target is probably going to appear on the left side. And then on a minority of trials, the target appears at the cued location. So the rational optimal way to do this task is to interpret the cue and then shift your attention to the opposite side, because that's probably where the target will appear. And if your attention is there, then you will get the benefits of attention on perception and do better at the task. And the beauty of this anti-cueing task is that you can shift between involuntary and voluntary attention simply by changing the temporal interval between the cue and the target. So in this case, if the cue is on the left side, if there's a very brief interval, that's long enough for involuntary attention to be captured, but it's not long enough for voluntary attention to be shifted to the right. And so attention is going to be on the left side when the target is shown. And that is the wrong side of the majority of trials. So even though the participants know the target's probably in the right, there's not enough time for them to shift their attention to the right. And so only the involuntary attention effects are evident here. And so in these 20% of the trials where the target is on the same side as the cue, the participants will do better. And so we replicated this classic finding. So this is a response time task, just pressing a button as quickly as you can to indicate the direction of tilt of the target. And so when the target and cue were on the same side, reaction times were lower. So this is the benefits of involuntary attention. Attention is captured by the cue. The target appears there right away on 20% of the trials. And so that's attention is in the right location for those trials, and performance is better than when the target appears on the opposite side. There's this extra shift of attention that's needed there. And so the cueing effect, is just the difference between these two, between the opposite side and the cued side. And so it's negative, indicating there's a benefit of the target appearing at the cued location. So not changing anything about the task except the timing. Now we use longer intervals. This allows participants to have enough time to shift their voluntary attention to the opposite location. And so here again, the cue is capturing attention to the left, and that's where the initial involuntary attention is. But then with enough time, attention can be shifted to the right, which is the most likely target location. That's going to be helpful on the majority of trials now. And it's going to be not helpful on the minority of trials for which the target is at the cued location. So we should see a switch in the cueing effect, and sure enough, we do. So here's the same data from before with the short interval. The longer interval, you see this big cueing effect. So subjects are doing better at the opposite location where their voluntary attention is directed compared to the cued location. We can also measure a cueing effect here, and it is positive in the opposite direction. So again, all the stimulus and task conditions are the same except for this interval of time. And now we can look at the effects of acetylcholine in performance of this task. So it's, again, a double-blind placebo-controlled design. Blue is placebo, red is donepezil. We see no evidence for an effect of donepezil on the benefits of involuntary attention on perception. And we see a small effect specifically at the opposite location for voluntary attention. And this is, again, an improvement in performance. And for all of these tests that I've showed you a drug effect, that the effect size is small. And I would say that's kind of by design. So we don't want to give large doses of donepezil that are completely changing the nature of cholinergic signaling in the brain. And so we actually use the smallest dose that's used in treating Alzheimer's patients. But for Alzheimer's patients, they're taking it every day for months or even years. We're just giving a single dose, and it's actually not subjectively perceptible. So we always ask participants to guess which day was the placebo day and which was the donepezil day, and there's always a chance in these experiments. So it's a very subtle shift in cholinergic signaling, and that's what we want. We want it to be physiologically similar to what acetylcholine is normally doing, and just changing it enough that we're able to determine what the effects are. And so all of these effect sizes are small, but again, it's kind of extraordinary that a systemic drug of any kind can improve performance on these demanding tasks. And so that's what we see again here, a selective effect on voluntary attention, but no effect on involuntary attention. So that's just shown here. But now at the cueing effect, there's no difference between placebo and donepezil for involuntary attention at the short SOA, and for voluntary attention at the long SOA, there's this enhancement. So donepezil has a selective effect on response time that causes an increase in the effects of cueing. So if the cueing effect is basically the benefits of attention on perception, that is what is enhanced for voluntary attention with acetylcholine. All right, so bringing it all together, I remember at the very beginning, we looked at the effects of donepezil on visual cortical responses in early visual cortex. We now brought attention into those kinds of measurements. So the stimuli are very similar as they were before. It's again 10 seconds of this contrast reversing checkerboard, alternating with 10 seconds of a blank period. But now there's an attention component. Now participants are maintaining fixation, and they're trying to detect these very small contrast decrements that occur at random times within the stimulus presentation and at random locations. So they really need to distribute their attention over just half of the annulus and ignore the other half. And they're counting silently the number of times the target is presented. So they need to maintain their attention throughout the 10-second period to do the task properly. So now we can look at the brain's response to the half of the stimulus that is attended and the half of the stimulus that's ignored, and compare that to the passive viewing case where there's no attentional demands. And this is again the binarization. So red means an excitatory response to the stimulus, blue means a inhibitory response. And so this is now a two-factor design. It's either passive viewing compared to attention, and then placebo or donepezil. So placebo is here at the top, and the passive view is here on the left. So if you look at the left side, this is basically the same result I showed you before, that increasing acetylcholine levels reduces the excitatory spatial spread of these responses. But you also see, if you compare the top row, the two images in the top row, attention is also reducing the excitatory spatial spread. And it's well known that attention increases the amplitude of visual cortical responses. But even so, even though the amplitude of the response is greater, there's less visual cortex that is showing that excitatory response. And then they seem to be roughly additive. So the effects of attention and the effects of acetylcholine are working in the same direction and do not occlude each other. They actually sum. And that's shown here. This is just a reduction in the amount of excitatory spatial spread compared to the baseline of passive viewing and placebo. Attention reduces spatial spread, so does donepezil, and then the combination of them reduces it quite a bit. So putting it all together, we found that cholinergic enhancement reduces the excitatory spatial spread of visual cortical responses with fMRI. It reduces the strength of surround suppression, so enhances performance on the surround suppression task. It benefits perceptual learning, and it increases the benefits of voluntary spatial attention on perception. And then attention can act like acetylcholine in that it also reduces the excitatory spatial spread of visual cortical responses, also enhances perceptual learning, and it increases the benefits of top-down attention. And so the way we're thinking about this is not that acetylcholine and attention are basically just the same thing. One is a psychological construct and one is a neurochemical construct. I mentioned the projections of the cholinergic axons in the cortex are very diffuse and broad. And so it's hard to imagine how you could get any kind of real spatial selectivity with those kinds of projections. And so going back to the Hasselmal model, the idea is acetylcholine is just moment by moment regulating how much an organism is oriented towards the environment in a feed-forward mode versus oriented more internally in kind of consolidation and suppressing information from the environment. And that the very rapid kinetics of the cholinesterase inhibitors are consistent with this, that there can be really good temporal regulation of acetylcholine levels that track the kind of behavioral demands or the overall state of the animal or person. And then attention is where you get the spatial selectivity. So these top-down projections are much more spatially precise, and they're indicating what aspects of the environment are important. For spatial attention, what locations, for features or objects, what elements are being highlighted by attention. And so attention is providing that selective mechanism, the filtering, in combination with acetylcholine, which is allowing, it's kind of a gating mechanism that is allowing more or less sensory information through at any given time. And so from that perspective, you can see one's kind of got the spatial precision, one's got the temporal precisions, and they could act together in the way that is consistent with what we found here. So I'd like to acknowledge all the wonderful people that contributed to this work. So Amitai Shenhoff was very employee number one in the Silver Lab at Berkeley 20 years ago. He's actually now a Berkeley professor as well. I'm sure a lot of you know his work. This was started when I was a postdoctoral researcher with Mark Esposito. Ariel's was our first graduate student. He's now at the University of Washington, headed for the University of Toronto. Sorry, so they did this. Amitai and Mark were collaborators in the spatial spread experiments. The perceptual learning was part of Ariel's dissertation research. All of the behavioral measurements and the effects of cholinergic enhancement were done by Anna Kusevcheva, who's now at Toronto, Summer Shirmata, who's now a teaching faculty at Northeastern, and Katerina Graton, who's at the University of Illinois. And then the NTQing involved Ariel and also Ayla Landau at Hebrew University and UCL in London, and faculty colleagues Bill Prince-Medal and Lynn Robertson. These are the U.S. government agencies that supported this research. And before I open to questions, I just want to say a little bit about a new research endeavor that Seung-Eun referred to a bit. The UC Berkeley Center for the Science of Psychedelics. A lot of the questions that we're asking here are kind of the flavor of the acetylcholine experiments I described. We're using psychedelics as a tool for selectively enhancing activity in certain serotonin receptor subtypes to better understand the connections between the mind and the brain and conscious experience. Psychedelics have really interesting and somewhat consistent effects on visual perception. There's also a lot of evidence that psychedelics together with psychotherapy are useful for treating a variety of mental health disorders, but we really have no idea how or even how they act in the brain. And so we're doing basic mechanistic neuroscience research with psychedelics in human subjects, together with brain imaging and behavioral measures to understand the mechanisms of action of psychedelics, but then also, just like we use acetylcholine to make inferences about mechanisms of plasticity and attention and so forth, we can make inferences about predictive coding and other perceptual phenomena in the brain by seeing how psychedelics alter perception and alter brain activity and the links between them. So thank you for your attention, and I'm happy to take questions.